Hello everyone, I'm Dr. Belhadj Benyahia Fayza. I'm from Department of Botany, the National Higher School of Agronomique, Algiers. So I'm here to present a part of our PhD doctorate. So the thematic it was intitulated to the Trichoderma atroviride-mediated activation of antioxidant defenses system to enhance wheat resistance against Fusarium crown root. So as an introduction, the wheat crop is the very important crop worldwide. It covers the largest cultivated area than many other crops. Unfortunately, these crops have some content like biotic content and abiotic content. As a biotic content, we have the disease of Fusarium crown root. This disease is very dangerous and dispersed in the worldwide. Is globally dispersed in area which have a higher and drought and temperatures. This disease can get the very high level of yield losses. So in the United States Pacific Northwest, they get the losses achieved 61% in winter wheat. In Australia, the losses achieved $97 million in wheat and barley. So this is not only affect the production or the production crop, but also can affect the human and the animal health by producing the mycotoxin, which these mycotoxin are cancerogen. So as management to this disease, there is many practical and using resistance varieties. There is also here we focus to the pesticide or fungicide, which are chemical products, chemical used to manage this disease. This kind of management is efficacious, but only in the early stage of the wheat growth cycle, and their use also pose harmful effect on human and animal health. For that, the researchers they focus the studies to use some biopesticide like bacteria, fungi, etc. Here we focus to use Trichoderma atroviride, which is a fungi which have a benefit to the plant, not only to protect a plant for against pathogens, but also to get the good nutrition to the plant. So to as a consequence, enhance or induce the growth of the plant. We know that in the defense of the plant against a pathogen, there is an antioxidant system. The role of antioxidant system is to maintain the equilibrium between the reactive oxygen species in the plant. Why? Because the reactive oxygen species like hydrogen peroxide can affect the health of the plant. It means the plant can get toxic if the level of this reactive oxygen species are past the limit. So to avoid oxidative stress. For that, the main objective of the study is to investigate the defense response induced by Trichoderma in 3 wheat varieties. So we want to get a comparison if the induction or amelioration of the reactive oxygen species in the antioxidant is linked with the plant genotype or no, and to focus on antioxidant enzyme, the induction, the antioxidant enzymes and polyphenols by Trichoderma. So as material we used here two genotype durum weight like as Waha and Vitron, and breed weight as like Ain Abid. As pathogen, we used isolate previously tested in wheat, which get a high level of aggressivity. And as antagonist, we used Trichoderma 13, which get previously the high levels of protection in many interaction plant pathogen. So here the seeds coated by antagonist suspension spore suspension, we cultivated in the pot as 16 seed by pot, and each seed has maintained in contact with gellose disc of the pathogen, 6 millimeter. So after you know that we had 4 treatment, a treatment which plant without any inoculation, the second treatment which is which was plant plus pathogen only, the third treatment it was plant plus co-inoculated, means pathogen and the antagonist. The third one it was the fourth one it was the plant inoculated only by the antagonist. After 15 days we get the samples from pots and stored it in minus 80 degrees Celsius. After that we can resume the technique or the method to get to detect or to estimate the level in each treatment. So in the first we do the grinding in each parameter, means for example for peroxidase and the polyphenol and catalase. So the grinding we using the buffer specific of each treatment, each parameter. After that centrifugation. After the centrifugation we did the assay, means in the polyphenol we used the Fullen method, the Fullen Sicato method. The calibration it was by using the Ecole. Peroxidase activity we used the method of Ecole and Ecole. The calibration we used the Ecole and peroxide hydrogen or hydrogen peroxide. For the catalase we used the method of Beers and Caesar. The calibration it was by peroxide hydrogen, and the detection and the determination it was by using UV spectrophotometer. Each parameter had the wave specific. As a result here, the main finding we found that there is inducing of peroxidase and catalase and polyphenol, especially in here the compared with the plant inoculated. This is here the plant inoculated only by the pathogen compared with the control. So we marked that Vitron had the diminution in the antioxidant system here in the leaves and in the basal parts. So can achieve minus 63% in leaves and minus 50% in basal parts. The polyphenol content when we compared with the co-inoculated plant compared with the inoculated only by the pathogen plant, we observed that there is induction of the antioxidant compound here for polyphenols can get achieved 1,400% in Waha genotype, especially in basal part. The peroxidase activity, the plant co-inoculated compared with the plant inoculated only by the pathogen, we observed here there is induction in the basal part, can get peroxidase 393%, So for the catalase activity, the same treatment co-inoculated plant compared with the inoculated only by the pathogen, we observed it was induction in basal part can achieve 60-40-100%, and in the leaves achieved 788%, especially in the Ain Abid genotype. As conclusion, the antagonist Trichoderma significantly enhanced antioxidant defense in wheat. A strong addition it was in Ain Abid and Waha. This result highlights a variety and genotype dependent effect on the response to the Trichoderma atroviride isolate. This result can be used in the wheat breeding program for varietal selection. And thank you.